here this long. My talk is an overview of the work that we have done over the past nearly 4 years in the assisted evolution treatment with algal symbionts sub-project. But before I start, I'd like to pay my respects to the traditional owners of the land on which we meet today, as well as to the traditional owners of the countries where the work that I will be presenting has been carried out. And I also like to point out that the work was a team effort, and all my team members are listed here on the slide. And we have a very diverse team consisting of research scientists, research technicians, as well as PhD and master's students. Now, you probably all know that corals form a close association with microalgae. They're called zooxanthellae or Symbiodiniaceae. And the corals are nutritionally dependent on these algae because the algae fix carbon and they translocate a large part of the carbon that they fix to the coral host. So despite the fact that the corals can also feed heterotrophically, they depend on these symbionts. Unfortunately, during summer heat waves, this symbiosis breaks down and the symbionts are lost from the coral tissues, and this is what we know as coral bleaching. And given that climate models predict that coral bleaching events are increasing in frequency and severity, if we can enhance the upper thermal tolerance limits of corals, then we may be able to buy some time for coral reefs until we reach a carbon neutral economy. Now, previous literature suggests that this may be achieved by manipulating those algal symbionts. And this is because variation in coral heat tolerance is largely determined by their symbiont communities. And also, heat-sensitive and tolerant symbionts exist in nature. This slide here was taken during a natural bleaching event on the Great Barrier Reef. And the two healthy Acropora mellipora colonies in the front were dominated by symbiont species in the genus Durasdinium, called D. And the severely bleached individual in the back was dominated by a cladocopium species called C. And we heard that already in Andrew's talk this morning, that these sort of patterns where there's variation within a coral species, variation in bleaching tolerance that is underpinned by the symbiont communities, has been observed across the world, on coral reefs across the world. However, on the Great Barrier Reef, and that sort of a little bit goes against Andrew's findings, we do very commonly see that naturally heat-tolerant symbiont species translocate a lower amount of nutrient to their host than the more sensitive species. And this manifests in a trade-off between heat tolerance and traits such as coral growth rates and reproductive output. And also, even corals that harbor the most tolerant symbiont species are still observed to bleach during extreme summer heat waves. So the aim of this project was to try and boost the upper thermal tolerance limits of the algal symbionts, and in this way also increase the thermal tolerance of the coral host once it establishes a symbiosis with these symbionts. And the way we went about that is through experimental evolution or laboratory selection of cultures of these symbionts. So we can isolate the symbionts from corals, and many of these can be brought into culture. And once we establish enough biomass, what we typically do is that we subdivide them into multiple replicates, and then some of those are maintained at ambient temperatures. We refer to those as wild-type cultures, whereas other replicates are exposed to stepwise increases in temperature, and we refer to those as heat-evolved. And the rationale behind this is that as the cultures grow and they go through their cell divisions, random mutations will occur in the genomes of these algae. And by chance, some of these mutations will be beneficial under the elevated temperature, and so these mutations will spread through the cultures. And what we expect that you will end up with over time, and this takes several years, is that that genetic differences accumulate between the wild type and the heat-evolved cultures. And also, different replicate cultures in each of these treatments will be different because they are independent evolutionary units. So, by chance, different adaptive mutations could occur in these independent replicates. And when we test these cultures, we indeed see that almost all of the heat-evolved cultures have higher heat tolerance compared to the wild type. When we then introduce them into coral, we see that only a subset of these heat-evolved symbiont cultures confer that enhanced tolerance on the coral host. And this sort of speaks to that, the independent evolutionary units that each of these replicates are. Now, it's relatively easy. We like to test these symbionts in all life stages of coral, so larvae, recruits, and adults. And it's relatively straightforward to get them into the early life stages that do not yet have the symbionts, that are aposymbiotic. But it's a little bit more challenging to introduce the symbionts into adult corals. And to achieve this, we've used a chemical bleaching method that was originally developed by Jan Matthews, who you just heard, for sea anemones. And we optimized that method, and what we do is we expose fragments of adult corals to repeated cycles of mantle or combination of mantle and diuron. And through this process, we can remove over 98% of the native symbiont community. Once the corals are bleached, we expose them to symbiont cultures, and we then just wait for the fragments to repigment, so to reestablish that symbiosis. And this whole process takes about 3 months. And we have shown through this method that we are able to introduce symbiont cultures into a taxonomically diverse range of coral species, including the ones that I've shown here. So it's quite a useful method. Now, because we cannot remove all of the native symbionts, when these corals recover, they typically recover with a mix of their original native symbiont community as well as the inoculum, so the heat-evolved culture. And I illustrate that here based on an example from the coral Galaxia fascicularis. So in that graph at the bottom right, each bar represents a coral fragment. And what I've plotted there is the result of a genetic analysis where the different colors represent different genetic variants, either of the heat-evolved symbionts, the inoculum, in sort of the orangey to green color, or the native symbionts in pink and blue. And you can see that all of the fragments recovered with a mix of the two groups of symbionts at different relative abundances. Now, the good thing was, even if some native symbionts are present, but we have some heat-evolved symbionts within that community, we still see the positive effects of that heat evolution. So we still see enhanced tolerance. And I summarize that in this slide. So we've tested our heat-evolved symbionts in larvae, recruits, and adults. And in all instances, we saw that the associating with heat-evolved symbionts enhanced the heat tolerance of the coral. And what was even better, we saw that it didn't compromise growth rates, as we often see on the Great Barrier Reef with the naturally heat-tolerant symbiont. So we're pretty excited about this, but you have to realize that all of these results were obtained in the lab, and they require validation in the field. Fortunately, we have a small field trial on the way, and I say it's small because at the moment we only work with one coral species, Patijara didalia. We have adult and recruits out in the field, and also this is only at one site, so it's quite preliminary. But we are obtaining data that will allow us to assess whether we meet our criteria of success. We set ourselves a number of criteria. And the first one, of course, is the fragments with the heat-evolved symbionts must have higher heat tolerance compared to the native corals in the field. But while that is true, we also acknowledge that the thermal tolerance phenotypes are likely to vary with environments. So the way we have defined this first criterion is corals harboring heat-evolved symbionts alone or in combination with native symbionts have thermal tolerance levels that are never lower compared with native corals and are higher at some reef habitats and sites because, as I said, we expect it to vary with environmental variations. And secondly, for an intervention to be economically viable, it has to provide the benefits to the coral reef over long periods of time. And therefore, the heat-evolved symbionts need to persist within these target corals. If we lose them, say, within a couple of months, then it's probably too expensive to keep repeating this whole procedure. And then thirdly, and this speaks to the scale of the intervention, we find that it's very important that these heat-evolved symbionts can make their way into the environment and then be acquired by other corals on the reef. During this field trial, we are sampling the sediment, the water column, and a lot of corals around our tables to see if we can detect the heat-evolved symbionts. But we already have some lab evidence that suggests that this horizontal transmission, this transmission from coral to coral of heat-evolved symbionts can occur. So what we did, again, we worked with adult coral fragments that had a small amount of heat-evolved symbionts in their tissues and also some of their native symbionts. We placed them into small containers, and then after 24 hours, we collected the surrounding water. Even healthy corals always expel symbionts. They do that to regulate the densities in their tissues. We filtered the water to concentrate the symbionts, and we assessed their health. And we also conducted genetic analysis to see which species were actually expelled. Now, about 80% of the expelled symbionts were healthy, and we found that the heat-evolved symbionts were expelled at percentages that are proportional to their abundance in the in-hospite community. So there was no selective expulsions, and they were expelled along with the native symbionts. And then secondly, using a custom designed sort of raceway system where we expose bleach coral fragments to different sources of symbionts. So I quickly explain it to you here. In the outer lanes, we have a couple of controls, and in the inner lanes, on the left, we have coral fragments that harbored about 5 to 12% heat-evolved symbionts. And then downstream of these donor fragments, we had bleached recipient fragments. And we found that about 4.5% of the recipient fragments took up the heat-evolved symbionts. Well, that is low, but the number of cells in the water would actually have been quite low from these donor fragments. So at least it's evidence that these symbionts can be transferred from one fragment to another. In the culture control, in the next lane, where we released algal cells over 8 hours each day, more than 50% of the recipient fragments had acquired the heat-evolved symbionts. So that is evidence, at least in the lab, that this horizontal transfer can occur. And this takes me to my last slide. So this is a cartoon of pretty much the inception of an intervention and the various stages that you go through, the path to implementation, if you like. And so we are in step 4. So in the field trial, we already done the design of the experiments. We've done the lab experiments. We obtained a regulatory approval and TO consent and things like that. So now, with a small field trial that will provide us some data to assess not only the benefits, but also the ecological risk of this intervention, and then hopefully that will allow us to begin larger scale field deployments, so at different locations, multiple species. And hopefully that will allow us to come up with better risk-benefit assessments. And I would think that that will allow us to then make recommendations as to whether we feel that this intervention should be implemented or not. If it is implemented, then we can think about upscaling, involvement of industry, and even maybe training to other nations where this intervention could be considered. And just along with this field work, some of the things that we're focusing on is expansion of culture collection is, of course, very important. Most of our cultures come from the central Great Barrier Reef, and we need to expand them to other regions. We're working on different culture methods to speed up that culture, to create biomass quicker. Optimal, better genetic markers to distinguish between our wild-type and heat-evolved symbionts for genetic monitoring in the field. And also we're thinking a lot about deployment strategies, whether we actually need to deploy coral fragments harboring those symbionts, or whether we can simply inoculate the sediments or the water column. And I'm going to just leave it at that so that you can go to lunch.